Massachusetts Department of Health, where she's the project director at the New England Pathogen Genomic Center of Excellence. She's also an investigator at the Division of Infectious Diseases at Brigham and Women's Hospital. She's interested in integrating pathogen sequencing data with epidemiological information to track the spread of infectious diseases, including optimizing the number of samples needed for effective pathogen surveillance. Shirlee received her PhD in systems biology at Harvard University and completed her postdoctoral fellowship at the Department of Epidemiology at Johns Hopkins Bloomberg School of Public Health. Today, Shirley's going to talk to us about sampling design for genomic epidemiology. So I'll go ahead and turn it over to Shirley. Thanks again for joining us today.
Great. Thank you so much for having me, for inviting me. I love talking about sampling design. I think it's a really exciting, I don't know, field of where we are in genomic epi. So I'm going to share my slides that I'll talk through today. I just want to encourage people, if you have questions or you want to discuss anything, please feel free to stop me. We don't have to wait till the end for questions. I think that there's a lot that we're still learning about sampling, so I think this can be a free discussion session if that's desired. Um, all right, let me see if Teams is going to cooperate with screen sharing. How does that look?
Great, we can see you.
Okay, perfect. And all right, so as stated, I'm going to talk about sampling design for genomic epi. I know that this is a genomic epi seminar series, so you probably have lots of talks describing genomic epi for public health. Obviously, I haven't been here before, so I don't know what you've seen already, but I imagine that you've had some talks looking at different kinds of ways to use genomics to cluster, like something shown on the left, or looking at phylogenetic trees, which I think is really exciting. Sorry, let me just make Teams. It's like in a weird corner of my screen. Let me just fix this really quickly. All right. So anyways, hopefully you've seen like this sort of diversity of examples, which I think is super exciting, because I think it shows that we're kind of in this new era of genomic epi, maybe slightly different than where we started a few years ago. One where it's being used maybe a little bit more to answer crucial public health questions, but also more importantly, one where we're not just generating sequences from whatever samples we have available, but actually thinking through how we might want to like prospectively design a genomics-based study. And so I think that that's what I want to focus on today, is some of the ways that I've been thinking about study design, some of the tools that are currently available to help us with study design, because I do think that this is sort of a new aspect of genomic epi compared to where we might have been a few years ago. So I'm going to start by shamelessly borrowing a concept I took from Allie Black. For those of you who know her, she's an epidemiologist in Washington, which she presented at AMB Days back in 2023. And she presented this idea of a whirling vortex of analysis, which I think is just a great metaphor. And this concept, I actually think, goes back a bit. I did some digging to find out who actually came up with this. I think Ali saw it on a talk by Lance Waller. He quotes somebody, John Richardson. I don't really know who to credit for this, but I think it's great. But I think it applies really well to genomic epidemiology, because it means that we really need to make sure that we don't get distracted by this like new exciting sequencing technology, and we can really focus on the question. So starting with like, what is the question we want to answer with our sequencing data? What's the problem we're applying it to in public health? But I think crucially, actually going like around the whole vortex of making sure that we're always coming back to the question. So we have the question, we have the data, we identify the data that we need to answer the question, then we figure out what data we can actually get. And based on that, we might actually have to tweak our question given the data we have available, since we might not be able to answer our original question. And I think this is a really important framework, because Historically, I think with genomic epi, we've kind of skipped straight from box 1 in the top left to box 3 in the bottom right, which is we have this question or problem, and then we have whatever sequencing data we've been able to generate, and we just move forward with that. And we really haven't had the opportunity to really think about, well, what data would we like to have? How would we design a study that is specifically to answer the question that we started out with? So I think this is the part that is changing, hopefully, is starting to change, and what we'll focus on today. So I don't know if we needed that intro. I feel like you all probably are familiar with these concepts, but I just really like this framing, and I'll come back to it a few times. So in the spirit of starting with a question, I'm going to start with two common types of questions that we can answer using genomic analyses, which hopefully are relatively familiar to this group. The first is questions related to individual transmission. So things like who infected person A, are cases from blue and the orange groups linked? Are there separate introductions? Are there clusters? Things related to how individuals are getting sick with a pathogen infection. And I think these are really important questions because how we target our public health can be really different depending if we have a scenario like this, where we have all cases are linked to the same index, for example, versus something on the left, which is the same 5 dots linked in a very different way. So that's sort of one kind of question, is how do we design studies that can figure out if we're dealing with the scenario on the left versus the scenario on the right. But then there are also a different type of question, which I think of as more population surveillance level questions. So things like, is a variant increasing in a population over time? Are there co-circulating variants? When was a strain first selected? I think these are the questions that a lot of us dealt with when applying genomic epi to COVID, really thinking about different variants. And so I think I'm going to start here in terms of thinking about sampling design specifically for these kinds of questions, and then I'll come back to the individual questions in a few minutes. So I want to start with, as I said, a specific question we want to answer. And so I'll just take one of the examples that I just started with, which is, at the point in time indicated by the arrow on my slide, are we detecting all variants of concern in a population? So are we seeing all of them, or are there some variants that we're just not detecting? And how do we make sure we have a sample that captures everything? So I want to call your attention to two parts of this sentence, which I think were very subtly bolded on my screen. One is that it's focused specifically on detection of variants. And then the other one is focusing on the population. And I think that sometimes when I think about sampling, I get a little carried away by the math and the ideas of it, and I forget to define what is the population at hand? Are we talking about a whole country? Are we talking about a state? Are we talking about a specific healthcare facility? And so I think this kind of all goes into the kinds of calculations and tools I'm going to talk about in a minute really only work when we have really specifically captured the question. So said what is the population we're looking at? What is the very specific question of detection? So I just wanted to throw that out at the beginning. So once we've defined this super specific question, we're thinking about detecting variants, and we've defined our population. We could say the state of Virginia, for example. I think there's two sort of sub questions we have to answer, which is one, how many samples do we need to answer this question? And two, which samples do we need to answer this question? There are lots of tools currently available for the first, so the how many. Some of you may be familiar with some of the tools that came up during the height of COVID that were used to kind of give rough sample size estimations or right sizing estimations for detecting variants specifically. So there was a great tool on the University of Texas Modeling Consortium, guidance put out by Europe CDC, a few different papers that came out. And then many of you may be familiar with the APHL influenza right sizing online tools. If you're not, they're great. And all of these tools sort of do the same basic thing, which is there's sort of this underlying probability theory of how many samples of the ones that we're like detecting in the lab do we need to make sure that we're capturing in our sequencing to get all of the detection of different variants. So in this example here, we have this pool of detected infections. We have 3 orange ones and a whole bunch of blue ones. And the question is, probabilistically, how many of the detected pool do we have to pick out and actually sequence in order to make sure that we capture at least one orange one? Because there are scenarios where you can imagine if we've only taken 3 from this detected pool, like the chances that we get an orange one is pretty low. And therefore, that sample size isn't going to effectively answer our question of are we detecting everything? So there's pretty straightforward tools to do this. All those calculators I just showed do that. But I think something that, oh, and I guess I had a slide explaining this with specific percentages, showing that, there are a lot of cases where you might not get any orange ones. But I think the caveat to a lot of this is that, and when you use some of these calculators online, they come with these like bias warnings that say, you know, all of these probabilistic tools assume that you are sampling representatively from the population you're looking at, which is obviously not true most of the time. So something that I have worked on in the past is thinking about what happens upstream of your pool of detected infections. So like what's upstream of what you have in the lab that you could actually sequence and look at, which is your whole world in your population, whether you've defined that as Virginia or the whole country or a specific hospital, it's all the infections that exist. And I think that we all know that there are some processes that kind of happen on this arrow on the left that might change how many orange variants we actually have that show up in the lab. So here's our pool. In my toy example here, 20% of the infections are of the orange variant, but something is happening here, which means that by the time we actually get to the lab and we have samples that we could sequence, only 10 of them are orange. I imagine that this group could come up with a bunch of reasons why that might happen. A few that I came up with were if the blue and the orange variants had different symptomatic rates so that you're more likely to be testing one of them than the other, which means one of them is more likely to show up in the lab than the other. You could imagine that the tests that you're using are maybe more sensitive for some variants or others. I think the gene dropout in COVID was a really good example of this. So there are lots of reasons why you might have that sort of inherent change already upstream of what samples you're trying to sequence. And I think I have on the next slide just like a very concrete example of the asymptomatic one, where if you imagine, for example, that the blue variant has a higher testing rate for whatever reason, because it's more symptomatic, they're more likely to get tested, then that's going to end up with something like what we just saw, where blue is overrepresented in your pool of samples compared to orange. The good thing, though, is that we can actually adjust the number of samples we pick from this pool to sequence if we understand what kind of bias process is happening. So I and other authors have developed a R package that kind of accounts for these different types of biases. So basically says, let's start with the math that all these other tools already use and just sort of add on another layer of can we account for some of these common biases and adjust our sampling accordingly. So in the example here, you might say that actually you need a larger sample size in order to make sure you capture the orange variant because the blue variant is overrepresented. So there are scenarios where you actually might need a smaller sample size because the orange variant is more represented. So there's different ways to think about it there. So we put out on this website, we have kind of a GitHub IO page. We have lots of guidance for determining specific parameters, recognizing that these sources of bias, why I think they're easy to conceptualize are maybe not super intuitive to estimate, like in terms of putting number values to what is the asymptomatic rate or what is the symptomatic rate, and does it vary between variants? But I hope and think that they can be a useful guide for thinking about, well, what is the range of sample sizes that we might need? And so I think that this is like a slight improvement, just coming back to this vortex idea of skipping straight from question box 1 to box 3 and sort of trying to think like, well, what does our data look like? What does the real world look like? And how do we adjust? So I wanted to just make this a little bit more concrete by showing you how we're currently doing this at Massachusetts Department of Public Health, specifically for our COVID surveillance. So for example, we are currently not getting as many samples in the lab as we did during peak pandemic times. And so we've been having a hard time tracking variants. And one of the difficulties to getting those samples in the lab, I think, was sort of buy-in from our clinical providers in terms of like, well, why do I need to be sending samples? How many samples do you really need? Like, why are you asking me for 100 a week? That's not feasible anymore. I don't see that many cases. So we applied this methodology to try to answer this question of, well, can we get enough samples from our clinical providers that we're reliably detecting anything that shows up at 3% or higher? And we picked a confidence that seemed right to us. You could tweak it in different ways. Um, and using those bias calculations that I talk about, that upstream, like asymptomatic rate, things like that, we were able to basically make like two different estimates where we said, okay, in the worst case scenario where we have this like blue variant overrepresentation that we saw on the other slide, this is how many samples we need. But in the best case scenario where we don't have that kind of bias, this is how many samples we would need. And so even though we weren't able to accurately measure, what is the bias exactly that we're seeing in these samples, we were able to say, okay, well, we have a like worst and best case scenario, which we're representing here as like the half as likely to detect or the just as likely to detect. And then we can put a range on, this is how many samples we need per week to answer this really specific question about detecting variants. And we actually sent some numbers based on that out to all of our clinical providers. And we said, hey, we're actually not asking you for that much. Like, we really only need 100 samples a week from the state. So like, could you please do your part and send us 10 samples a week? That would be great. And it really worked. We started getting a lot more samples in the door, and I think our surveillance is going much more smoothly because of that. So I'll just flag that all of this is in that PhyloSamp R package that I mentioned, but there's also, if you're not an R user, we have a spreadsheet that does all the calculations, and some specific instructions are actually about to go on our New England Pathogen Genomic Center of Excellence website. So I can put that in the chat at some point, and maybe Alex, I'll flag to you when it's available if that would be of interest to folks here. So just a couple more notes on this population level surveillance question before I switch to the other individual question. I just want to admit that I really only talked about the first part of the question, which is the how many samples we need question. And I didn't talk about the which samples question. And that's because it's a lot harder of a question. I think it's related to that bias warning that I talked about earlier, that even though we have some methods to correct for known biases, such as testing rates, we're still making some underlying assumptions often about things that we can't measure, like how representative is it of like a geographic population, or are our samples well distributed over some other important variables. So I just wanted to flag that, like, I think that this is not quite answered yet. And I think is something to still keep in mind, so that even though, we said we need 50 to 100 samples a week, like that does assume that it's like well distributed over our clinical providers throughout the state, and all the other variables that we're not explicitly correcting for in our bias calculations. And I think that that's sort of an exciting place for this field and development to go, is thinking about other ways to incorporate other types of bias. So I think with that, I already talked about this. So I think with that, I will pause for a second And I'll just mention very briefly that I was talking really specifically about the detection question, but I think that there is a similar and related question of prevalence. So instead of saying like, I want to detect yes, no, is the orange variant here? It may be a question of, well, am I able to estimate its prevalence? So when I find that it's 10% in the population, is it actually 10% in the population? And the same framework that I just presented in that PhyloSAMP package answers that question as well. So I just wanted to maybe pause there for a second. I feel like that was a bit of a whirlwind in all things sampling for population surveillance, and see if anybody had any questions or comments before I switch over to the individual transmission aspect.
Question. It's not specific to the sampling, but more so like how did you share this information back with the clinical submitters? Like, did you share a report or have like a meeting where you shared this with them? I just find it interesting that this helped to get more buy-in when you were able to give them data or like numbers around like how their samples are feeding into the surveillance system.
Yeah, so we did both. So we had a meeting with like our state lab directors and all of the clinical lab directors around the state. And they sort of voiced this issue. They were like, well, you're asking us for 100 and I don't think you need 100. And what's the number we need? And then they also voiced that they wanted more information coming back to them. So like, even if it's not a like, we can't report like a line level diagnostic, like this patient had this variant, they want to know what the sequencing data is being used for. So to follow up with that, we sent out a memo to all of them that had this sort of really specific sampling guidance. It didn't go through all of the like what I just showed you, but it was just a, hey, to meet our targets, we need you to send 10 per week. And that is based on sample size calculations. And we're happy to follow that up if you need it. And then the other thing that we're actually currently doing is we've worked on a sort of aggregate report that goes back to individual institutions with sort of some basic variant information and how certain we are of detection based on these calculations. That answer your question?
Yeah. No, that's a really great way to, I think, to try to get more buy-in. Thanks.
Yeah. And it's nice because I feel like there's already so much standardized for influenza, right? Like we were able to really clearly point to the CDC guidelines, and then we kind of followed it up with like, and for our tracking in Massachusetts, this is what we would need for that.
There's a question in the chat too. I'm not sure if you're able to see this or not.
Oh, let me pull that up. Okay. How are you quantifying uncertainty in the parameter values? Do users provide an explicit? That is a great question. So in the parameter values in PhyloSAMP, we actually aren't asking for explicit parameter values as an input, but rather the relative values between variants that you're interested in. So instead of saying like the asymptomatic rate is 85%, it's actually asking for variant 1 versus variant 2. Like, is this asymptomatic rate 50% higher or 20% higher or something like that is really what's like underlying. So it doesn't really matter what the raw numbers are as long as we're able to estimate that sort of relative ratio. Understanding that that is hard to estimate as well, which is why I sort of presented the example of, I think in practice, how we're using this is a little bit more like, let's come up with the worst possible ratio we can imagine and the best possible ratio we can imagine and run the calculations for both of those and use that to give a range of guidelines for sampling.
Gotcha. Yeah. So just to follow up on that, I guess. So you're providing like single values. So the asymptomatic rate for variant X is this number, for variant Y it's this number, you're not explicitly, is that kind of assuming like a normal distribution for both of those, or does it not even assume any shape of that distribution whatsoever?
It's not assuming the shape of the distribution just because the way the calculations actually worked out, it's all really about the ratio between the variants on average. So yeah, I guess we are assuming sort of a bit of a distribution there. I'd have to look back and exactly what we were assuming.
Gotcha, thank you.
Yeah. No, it's a good question. I mean, because there is so much uncertainty in these values. Well, I can spend a few minutes moving over to the individual transmission question, and then should hopefully have some time for questions after that as well. Okay, so as a recap, we are shifting now from these like population surveillance questions to more of the who infected who type of questions. And I think it's important to understand these types of questions because it's so related to like the other application of genomic epidemiology, which is a little bit more the like cluster investigations, trying to understand what is going on in an individual outbreak. And I think it's quite common to do things like make phylogenetic trees to look at this kind of information. And I think it's really easy to look at a phylogenetic tree and pull out something like this, where it's like, oh, okay, samples B and C are really closely related. And it's really tempting to say, we think that there's a transmission link between them because of how they're showing up in this phylogenetic analysis. But I think there's a lot of cases where that's actually not correct. And what we actually have going on is something more like this, where B and C aren't direct links, but are actually linked by some unsampled intermediate, which I'm calling X here. And so I think this example sort of shows, again, the specific question that I'm trying to get at. So really trying to make sure we're like narrowing down on a specific question before developing the methodology, which can be summarized of are closely related pathogen sequences indicative of direct transmission. And I think this is relatively easy to to conceptualize in the sense of if you imagine an outbreak where you sample 100% of cases, if you see something like B and C like this, we know that they're direct transmission links because we know that we've sampled everything. And if these two look the closest related to each other, we're assuming that they are linked by transmission as well. But if we don't sample 100% of the cases, how many do we have to sample to get to the point where we feel relatively confident saying that these are actual links? And so again, I think we can split this up into the how many versus which samples question. And the which samples I think is like a little bit easier this time because we're dealing with a much smaller scale than in the previous. So in the previous, we were talking about like population level surveillance. So there's so many considerations. There's considerations about geography, considerations about making sure you're capturing certain groups, versus here, it's really just about like, well, we want to capture as much as we can about this outbreak that we're interested in. So I think in this case, the how many is the more interesting question. And I'll start there again. And so I wanted to just, again, hone in on the fact that we really do have to be really clear about these questions to develop a mathematical framework to go along with it. And so I wanted to pull out the phrase closely related because it's actually pretty vague, right? Like, does closely related mean direct link? Does it mean that they look similar on the tree? Does it mean a specific mutational threshold? So the first thing that we did when we thought about this question was try to narrow down what that meant, specifically by defining what we called a linkage criteria. So say, okay, if we can come up with a criteria, then can we come up with if it meets the criteria, how likely is it that they're actually linked? So I think a concrete example of this could be, let's say our criteria is 2 samples must be within 2 SNPs of each other. And so once we've defined that as like the threshold or the criteria, we can say if they're within 2 SNPs, what is the probability that everything we see within 2 SNPs is actually linked? And what is the probability that we are missing something in the middle? And so that's how we were sort of thinking about it. I think this is an area that like needs even more development than the previous one, but we do have a tool also implemented in PhyloSamp that builds out this method of saying, if you can define that criteria, can you calculate the probability? And I'll just walk through a quick little example of sort of what we're thinking about that. So again, let's assume dealing with SNP thresholds because that's a really easy criteria. So in this example, I am showing a SNP threshold of 4 SNPs. So saying that this is like in the light orange is the distribution of unlinked case pairs, and in purple is the distribution of linked case pairs. And so we're saying that, you could even just visually look at this and say, wow, okay, 4 SNPs really clearly differentiates between these different things. And we can actually calculate that and say, okay, the area under this curve, this is just like sort of a classic ROC curve. And we can say that looking at the specificity and the sensitivity, they're both very high. And we can, with this threshold, with a lot of specificity and a lot of sensitivity, distinguish between whether things are actually linked or whether they're not linked. Of course, this is nicely simulated data. It's not real data. Real data tends to look something like this, where this is actually data from a real mumps outbreak, where we had information about what cases were really linked and which ones really weren't linked by transmission. And we made this plot of genetic distance. Again, this light orange color is the unlinked pairs and the purple are the linked pairs. And we could play around with where we put the threshold, but no threshold is perfect, right? Like, I mean, we could put this threshold all the way over here at like 8 or 9 SNPs, in which case we would always be able to tell when something is linked, but we'd also get a lot of false positives. So things that are actually orange and unlinked would look linked because they're fewer than 9 SNPs apart. Or we could push our threshold lower, but then we would miss a lot of truly linked pairs. So this, we can play around with this. We can make different ROC curves and sort of figure out what is the best threshold. But even with the best threshold, we're going to have imperfect sensitivity or imperfect specificity at determining what is linked or not. And I think that's where the sample size calculations come in, right? Is like, what is the sample size that you need to get to a certain degree of sensitivity or a certain degree of specificity, depending on what your question is. And so that is what is in the implementation of PhyloSamp for this question. And there is also a tool that says, okay, if you know some basic things about your pathogen, like the mutation rate, like the generation time, we could actually simulate something like this and infer what we think could be the best threshold and use that to inform your sample size calculations. So again, it's not perfect, but it, I think, can provide some useful guidance on, are we even sampling the right ballpark of number of samples to be able to answer this question? And I personally think this is really important because I've seen so many studies that are like, oh yeah, these are close to each other on the tree. They must represent a transmission link. And it can be actually pretty shocking to see that like, you need a fair number of samples to be able to say something like that with a really high degree of specificity. And I think that that's exactly what's needed to split between these really different types of scenarios. I also just want to acknowledge again that like here, the linkage criteria that I defined is a genetic distance threshold, right? I was saying, okay, 4 SNPs or 7 SNPs, it's just a genetic distance. But that's a really simple criteria. I think the future of this field is thinking about other types of criteria that we could use to improve those sample size. So if you were able to make a tree and have something like clade structure or clade support or additional epidata that's layered onto the tree and have that be part of the criteria so that it's not just two SNPs, yes or no, and it's like a more informed criteria by other data you have other data you have available, then I think you could use that to modify your sample size calculations and perhaps need fewer samples to be able to say something more definitively. I just haven't figured out how to do that yet. And I hope that somebody will figure out how to do that in the near future, because I think that it could really help with some of these important questions. So that I would say is most of the content I have for today. I'll just summarize some of the takeaways that I think are applicable to both of the population level surveillance and the individual level surveillance. The first is it's like just always start with the question, which seems, I think, so obvious in the context of a talk like this, but in my mind actually is very often overlooked. I think it's really exciting to be like, great, we have like 200 samples, like let's see what we can learn from all these sequences. And I think that the answer, and I think the answer to that really depends on, okay, but like what are we trying to do? Like, is 200 enough to do direct transmission? Is it enough to do detection? Is it enough to estimate prevalence? Is it enough samples? The answer is going to depend a lot depending on what that question is. I think the other thing that to me is a big takeaway from all of this is making sure to consider both which samples and how many. I think it's really easy to say, okay, we need guidance. It's 100 samples, that's the answer. And something that I think we've really learned in Massachusetts is if we're not careful to think about the which samples, all of the samples come from the same hospital in the same part of the state and really are only representative of a very small fraction of our population. Acknowledging that this question is really hard to answer. But I think even just keeping it in mind has been helpful in terms of making sure that, we have geographic representation, even if we don't have specific math to back up the which samples aspect of it. And I think the other sort of big takeaway that I always think of is that these calculators, at least in their current form, are really to provide general guidance. There's never going to be something that's like, okay, if you have 100 samples, you're going to be able to answer this question with certainty. But I do think it's useful to get an order of magnitude. I think in Massachusetts, we were actually quite surprised that the number of COVID samples we needed to get at that 3% prevalence question was only 50 to 100. Like, I think people were assuming that we needed hundreds and hundreds of samples every week because that's what we were doing sort of during the peak of the pandemic, and seeing that, oh, it's only 50 to 100. And that means that each clinical provider only needs to send us 10 or 15 was really useful. And even if it's a general guidance, not a specific number. And of course, I think there's this whole element of sampling biases, but the more we think about them, the more we can account for them in our calculations. So I'll just end with a slide with links to some of these right sizing options. And I'll just reiterate that a lot of these are, our packages, but for pretty much everything I have here, there's either a spreadsheet or an interactive online app or both available, so that they can be implemented in lots of different settings with or without access to R. So I'll just leave this slide up and see if there are other questions. As I mentioned, we're actually hoping to put a lot more sort of examples of what we've done and instructions to do the same thing up on our PGCOE website coming pretty soon. And so if there are particular things that you would find like helpful to see there, please let me know, because we would love to put those up and make sure that this is optimally useful for anybody else who wants to take a look. So with that, if there are any other comments or questions, I'd love to talk.